everyone, this is Talkside brought to you by Waithira. Most people call me Carol Ndegwa. It is another week and another episode, and this week I'm bringing something different, and therefore I feel that it wouldn't be relevant for me to have it on the Spotify platform. So my listeners, you can just move on to the YouTube channel. It is still titled the Talkside Podcast, and watch it, like, comment, and share with your community. As I have mentioned before, I am a student at the University of Eldoret. I am pursuing plant breeding and biotechnology, and one of the requirements for me to fulfill before I can graduate is to have a seminars presentation, which is part of my first year's units, and I was able to present it during this past week, which marked the end of my first year. Really excited about that. And yeah, I would like to take you through what I presented, so let's go through this.
Carol?
Hello everyone. Yes, I am ready.
Okay, so I'll give you the right to move the presentation, and there you go. Karibu.
Okay, thank you. Following the lift of the ban last year on genetically modified organisms and the adoption of the organisms in Kenya, I thought it was good for us, or important for us, to be able to understand the regulations that are there for the GMOs in Kenya. So genetically modified organisms are products that are developed through modern biotechnology, and they normally contain a combination of novel genetic materials. Globally, 26 countries are already boosting their agricultural productivity using these organisms. And between 1996 and 2018, 2.5 billion hectares already are being commercialized using GMOs, and the approval of this cultivation and also the research is normally done on a case-by-case basis. And of course, there has to be a regulatory body that ensures that all this is done according to the book, and in Kenya, it is done by the National Biosafety Authority. Maybe first, I think it's important also for us to understand what is genetic engineering, and it is the manipulation of organisms, in our case, plants, using recombinant DNA technologies. Recombinant DNA technologies uses enzymes and laboratory operations to improve crops, and that is to improve crops for abiotic stress tolerance, disease tolerance, pest resistance, increase their yields, and so on. It is also important to note that some of the organisms that already in Kenya have been approved for such research. There's the bacterial wilt resistant banana. There's also the insect resistant pigeon pea. There's the stress tolerant cassava, and also drought tolerant maize. So there's already research that is going on, and continuing to understand on what genetic engineering is, there are methods that have been used to improve these crops. There's the indirect methods, where there's the use of the Agrobacterium tumefaciens, and the indirect methods, just to mention a few, there's electroporation and microinjection. To understand further, I thought to bring in a diagram that will enable us illustrate agrobacterium-mediated transformation with a case study of sugarcane. So this is a plasmid, and a plasmid is a genetic structure within a cell, and its ability to be able to replicate independently of the cell is what makes it useful for gene transformation. And then there's the plasmid normally has a certain region that contains auxin, cytokinin, and opine genes, and these genes normally have hormones that cause tumor inducing in certain plants. And for one to now start their program in gene transformation, they have to have identified a desired gene and isolate it from a source organism, and that they'll start by creating a cassette, and a cassette normally contains the target gene. It will contain the promoter and terminator genes that will enable the expression of this gene, and it will also contain a marker that will enable the selection of the desired gene. Once the cassette has been formed, it is normally now replaced to that region that had those genes of the auxin, cytokinin, and opine. So that region is normally replaced with the cassette that has been formed. Once it has been integrated in the plasmid, it is inserted inside an agrobacterium tumescens that will act as a vector. So the process moves on to through plant tissue culture. So they will now identify the crop that they want to improve, and through tissue culture they will get some, maybe let's say some leaf discs, cause some wounds, and co-culture both the bacteria that has now the gene of interest and the plant they want to, the target plant. So once during that culture, there's the integration of now the bacteria and the wounded parts of the plant. And then once that happens, they will now move to the selection stage. During the selection stage, it's again through tissue culture, and the media normally contains a biostatic. This biostatic prevents the growth. You do not want too much growth again of the agrobacterium. And the media also contains a selective antibiotic that prevents the growth of the untransformed tissues. So it is only for the selection of only those tissues that have already been transformed. Then you move on, you regenerate the plants normally using tissue culture from colors to shoot to root, and then you obtain your already transformed plants. So now we have understood how the research or the transformation processes takes place. So now we can move ahead to understand the regulatory frameworks around this. Sorry for the juggling of the words. I think during the sharing is when this occurred. So because of how the lifting of the ban and now the adoption of these organisms, there have been questions on what is the safety of these products on the human health, environmental health, and also on other animals and plants. And so Kenya has found it important to have a regulatory and institutional mechanism to enable the implementation of these technologies. And inside those mechanisms, there's what is called the Cartagena Protocol. I will discuss all this in the next slides. There's also the National Policy on Biotechnology Development and the enactment of the Biosafety Act in 2009. So we can start with the Cartagena Protocol. So Kenya is a member of the United Nations, and the United Nations normally have different treaties and different agreements. One of such agreements is the Convention on Biological Diversity, and inside the Biological Biodiversity Convention of Biological Diversity is the Cartagena Protocol. The Cartagena Protocol mainly focuses on the development of modern biotechnologies and to address the concerns regarding their safety. It was adopted in 2000 and its supplementation was done in 2010, and its supplementation was done by the Biosafety Act of 2000. The protocol is mainly to ensure the safety of these GMOs to, again, humans, environment, and also the animals. There's the Biosafety Act that was enacted in 2000. The Biosafety Act led to the establishment of the body called National Biosafety Authority. And the main objectives of the Biosafety Act is to facilitate research and minimize the risks that may be posed by GMOs, to also ensure that there's adequate level of protection during the development, the transfer, and also the handling of these GMOs. But most importantly is to ensure that there's a transparent and science-based and also predictable processes during reviewing and making decisions and also policies of these GMO products. I also mentioned about the National Policy on Environment, and this normally ensures that risk assessment tests are done to ensure that there are no toxicological, negative toxicological reports about the GMOs, social economic considerations to ensure that these GMO products are, during their development, it's cost effective, but it is also bringing profits to the farmers. Public participation, where we as a public are allowed to have our say or our opinions during these regulatory measures. And there's also the consultation from the regulatory agencies, as I've mentioned, as such is the National Biosafety Authority. So the establishment of NBA has 4 major regulations, and that is contained use, meaning that the research can only be done in certain facilities and certain laboratories that have been approved. Environmental use, this is done by ensuring that the field trials that are done under confined field trials. The export, release, and transit is also ensured to ensure that there's no GMO that has not yet been approved can go outside the borders. The labeling, there are some countries that have allowed labeling. In Kenya, I don't think they do label the GMO products yet. So the procedure followed during the release of a GMO, that is by the regulatory body NBA, is a researcher or a breeder will fill out a certain form and they'll submit it to the National Biosafety Authority, who will then ensure that there's accuracy and completeness in that form. And then they will be able to, through their laboratory tests or certain tests they do, they will check whether the product has a foreign genetic material, and then if it does or it doesn't have their certain protocols it will follow. The breeder is then advised to continue doing the national performance trials, the confined field trials, distinctive, uniform, and stability tests, and also laboratory tests. So in case the NBA has checked your product, and they are able to see that your product doesn't have any foreign gene, that means it won't undergo the CFTs. Gene edited materials, therefore, do not go through CFTs, and this is because they do not have any foreign material in them. So this is just further explaining those that have foreign material and those that do not have. So how are then NPTs performed? They are normally performed across the country to ensure that different agroecological zones are covered. At least 2 growing seasons are expected with a minimum of 3 replicates. The body that does these NPTs and most trials, multi-locational trials, is KEFIS. Once they have done all these tests, they'll compile a report, submit it to a committee, the National Performance Trials Committee, who will then check through the report, and they are able, if your material or your crop meets the National Variety Release Committee criteria, it is then recommended, of course, with support from other reports like the DOS reports. And then if it meets all the criteria, it is then gazetted and listed in the National Crop Variety List. The breeder is then allowed to multiply and sell their new variety. So confined field trials are specifically for the GMO organisms, and they are normally done, of course, to evaluate the expression of the traits and also to test for agronomic performance of traits. But most importantly is to check the biosafety data, because remember, there are all these questions on the safety of these organisms. So CFTs mainly is to check on the biosafety, on whether they are safe for environment, human, and also the plant. And also to ensure that the food tests are done to ensure that they are good for consumption. The guiding principles for CFTs, one of them is material confinement, and this is to ensure that all products are labeled. In case they are genetically engineered plants, they are labeled. All cleaning material used during the confined field trials are cleaned, properly cleaned. The material also gotten from these CFTs are also disposed appropriately. That can be done by burying, chemical treatment, or also incineration, which is burning. And also these CFTs are normally well gated to ensure that no individual is able to get in and get any material from these trials. Another guiding principle is genetic material. This is to ensure that there's no mixture of the pollen from these GMO products to other crops that may be neighboring at this CFT. This can be done through spatial isolation, where there's a certain distance between the GMOs and also the plants planted around that region. Temporal isolation is done. You are supposed to do it and timing so that you are late in planting the GMO crops, so that the anthesis period does not meet with the neighboring individuals. There's the bagging of reproductive parts. There's also early termination of the crop, removal of floral structures, and also creating pollen trap rows. All this is done to ensure that the pollen from these GMOs does not mix with any neighboring crop. There's the post-harvest restrictions. These are normally done after post-harvesting, and it is also to ensure that there's no crop or any material that is left behind after a trial has been carried out inside that CFT. This is done by banning all volunteer crops just before they start flowering, and also within that CFT, there's no same crop of sexual compatibility is planted in that CFT. Again, is to control any movement of these genetically modified organisms. Some of the challenges in the regulation and the release of these GMOs, there's a low public awareness, and people are not aware, and that's why they are also questioning about their safety and so on. And we, as people who are in the biotechnology department or people who understand science, are encouraged to have science communication so that we are able to disseminate all this knowledge to individuals, to also increase adoption, because we know of the importance that these GMO products have in increasing food security in our country. There's also different bodies driving the same mandate. So if a product has been presented for regulation and a certain body approves and a certain body and another body does not approve, then that brings about conflict. There's also political and economic interest, where you'll find that certain politicians will be talking ill about certain GMOs, and this is because they want to beat their opponents and so on. There are also individuals who would not want the GMOs to be in the market, probably, maybe because they are bringing a lot of competition to their products. These are some of the references. If you would like to understand more about the regulation of GMOs in Kenya, especially, you can log into the the National Biosafety Authority website, and you'll be able to understand more and also to see the products that are already in commercialization. Thank you.
Yeah, thank you, Carol, for the wonderful presentation. I can see that we are learning on regulation of GMO and
So that is the end of the presentation, and would like to thank you for listening to it. During the presentation, I do mention that the Biosafety Act was enacted in 2000. I would really like to correct that it was enacted in 2009. There were two questions that were brought up during the presentation, and one of the questions was on what dictates spatial isolation. I mentioned that spatial isolation is where there's a certain distance that is normally checked or required between a CFT and the neighboring crops, especially if it is a cross-pollinated crop. And the answer to that is that the distance at which a pollen is able to travel and fertilize a neighboring crop. The next question that came up was on the labeling of GM seed. I wanted to be sure of that, and so I went ahead to Google that, and Kenya has a law. It's called the Seed and Plant Variety Law, and it requires that all products that are produced or developed through genetically or genetic engineering are actually labeled. And I would like to quote that a statement that says that the labeling of seed derived from genetically modified plants shall be considered after they have undergone appropriate GM seed authorization and certification. And this is done by two organizations, and that is the National Biosafety Authority and also KEFIS. As always, guys, thank you for being here, and see you on the next one.